start. So, just a second. Ready? Yeah? Okay. We'll start the webinar series. So, good evening to all the participants who have joined us from India. Good morning to those who are joining us from US, and good afternoon to those who are joining us from the Europe. Welcome to this Consortium webinar series. As you know, we have these series of webinars by eminent scientists once in 2 months periodically, and we choose topics that are of relevance to the area of work that we are interested in. And today we have a very accomplished researcher with us, Dr. Chinmay. He is from Agarkar Research Institution, Pune. He is going to take us through his journey with Zebrafish and his experiences with this model system. And he's going to talk to us about a very interesting topic called heart regeneration and how Zebrafish is a very effective model system in this field, in the field of regenerative medicine. Before we do that, let me introduce the guest of today's webinar. We have with us our Honorable Vice Chancellor of the University, Professor Dr. Satish Kumar Bhandari. Sir is ever approachable, ever supportive, ever encouraging for all the activities of the institution, and for a research institution like ours, sir has always been quite effusive in his praise for NUCSER and his love for our institution. Although we are doing it at evenings, beyond the office hours, sir has kindly agreed to take part in this webinar. Thank you very much, sir. I will introduce our resource person in a bit, but before I do that, I would like to request our Vice Chancellor, sir, to give his opening remarks. But since we have a number of participants from outside Nitte and several other institutions, I would like to take this opportunity to briefly introduce our Honorable Vice Chancellor to the August gathering. Sir is currently serving as a Vice Chancellor, and he has been serving as the Vice Chancellor since November 2017. Sir has served in various administrative positions in his illustrious career. Now, before joining Nitte University, he was associated with KMC Mangalore for about 10 years, and then he joined as a professor and head of the Department of ENT in 1999, where he served in this position for about 13 years, followed by his elevation as a vice dean for a period of 5 years, and then as a dean of a medical college for another 5 years. Sir has held a long list of executive and non-executive posts, such as the president of Indian Medical Association of the South Canara branch, president of AOI Karnataka chapter, and Karavali branch. Sir has contributed immensely to the functioning of these branches through the conduct of several seminars and lecture series. He has been an active researcher himself. Sir has several patents in his name. He has obtained several external research grants from BRNS, Government of India. Sir has involved himself in many extension and social causes, and he has established the Mangalore Laryngectome Club at KSAP Medical Academy, and which is also a registered member of the International Laryngectome Club Association in the country. Sir has been bestowed with several honors and awards, the most prestigious being the Aryabhatta Award for Excellence in Field of Medicine by Aryabhatta Cultural Association in 2006, Distinguished Alumni Award from Manipal Academy of Higher Education, and Eminent Educational Award by National International Compendium, New Delhi. Most recently, he has been bestowed FRCS from President of the Royal College of Surgeons of England in October 2018. Sir has also been awarded the Fellow of the International Institution of Organizational Psychological Medicine. He's also been awarded Swami Vivekananda Excellence Award in 2019, Lifetime Achievement Award by Association of Otolaryngologists of India and Karnataka and Kodagu branch in 2019. Sir has over 72 publications, has participated in more than 100 national and international conferences, over 25 presentations in these conferences worldwide. Sir has also a patent on a mouthwash for oral radiation for radiotherapy patients, and which was published in 2020. Sir has also authored 2 books, one of which is totally different from his area of interest. This book is about rare medicinal plants of coastal Karnataka, and he has also published another book on ENT from in the local language in Kannada. So it is a great honor to have such a distinguished administrator, a researcher, and a scientist, an avid follower of research amongst us. Before we request Dr. Chinmay to present his webinar, I would like to request our Honorable Vice Chancellor to please give his opening remarks. Over to you, sir.
Good evening. Respected, very distinguished resource person, Dr. Chinmay Patra, Dr. Anirban, and other distinguished guests for participants in the webinar, and my colleagues from Nitte University, and the people who have registered for this very interesting webinar conducted by our prestigious institution, Nitte University Center for Science, Education, and Research, what we call NUCSER, under the seminar series of the Consortium of Rare Genetic and Bone Marrow Disorders of Nitte University. I congratulate Dr. Anirban Chakraborty for having this webinar with the distinguished faculty Chinmay Patra. It's a matter of pleasure to me to be part of this webinar, and the theme of this particular webinar is very interesting. I understand we are going to discuss about heart and various issues about the heart, particularly the ability of the heart for regeneration. As you all know, Nitte is committed for promoting research, and one of the most successful outcome of these efforts is our establishment of NUCSER, a state-of-the-art research institute established in the university to carrying out cutting-edge research in the field of biomedicine. And it's with the collaboration of various clinicians. We have got medical school, dental school, and pharmacy school, and other health-related institutions. And also, very distinguished research faculty have been enrolled in this prestigious institution. The large number of external research projects that the NUCSER faculty has managed to bring in, the foreign collaboration the faculty has brought, and the quality of research which is instituted by this institution, the kind of research publication that are coming out of this institution, and above all, the dedication and commitment of the faculty of NUCSER is a testament to the fact that NUCSER is performing exceedingly well the last 6 years. The Consortium of Rare Genetic and Bone Marrow Disorders, which was established with a seed grant from the university in the year 2018 under NUCSER, and it has got various research projects. The main aim is to delineate genetic spectrum of rare conditions. Today we have a very accomplished researcher and scientist par excellence, Dr. Chinmay Patra from Agarkar Research Institute of Pune, will talk about zebrafish and their ability to regenerate heart, and how this model system can contribute for better understanding of the process of tissue degeneration. I'm told that Dr. Chinmay has recently published a paper in a prestigious journal where he has shown the function of one particular gene in heart degeneration. Congratulations, Chinmay, for this wonderful work. Nitte has a very functional zebrafish facility under NUCSER. I understand it has got various funded projects also by mainly by ICMR, and some are related to COVID-19. Studies are related to COVID-19. In addition to this, there are a number of other projects that are also ongoing, mainly on developing disease models and genetic systems using, I understand, CRISPR model they are trying. In the year 2020, we upgraded this facility. We had little earlier we had a smaller setup, and but I'm very glad to tell you that last year or the year before that we upgraded our zebrafish facility, and we have imported the state of that facility, particularly from Italy. And I understand I was talking to Anirban. I understand now the facility provided by Nitte is as good as provided by renowned institutions like Indian Institute of Science, and even what Chinmay using in his setup is also similar to ours. That's what I understand from Anirban. The topic that Chinmay is going to discuss is very, very relevant, particularly country like India. As you all know, ischemic heart disease or the myocardial infarction and the stroke constitute almost 85% of the cardiovascular diseases. And according to WHO, India accounts to about one-fifth of the cardiovascular disease-associated deaths. It's really mind-boggling. Uh, even according to Sustainable Development Goals by United Nations in the year 2015, one of the proposed target is reduction by one-third of this premature mortality caused by cardiovascular diseases and other diseases like respiratory disease, illnesses, cancer, and diabetes by 2030. It is also one of the highest burden of cardiac disease in the world, and high prevalence of the five cardiovascular risk factors, including raised blood pressure, raised blood sugar, obesity, smoking, and overweight, is highly seen in our population. Although these are the various statistical data, and you will agree with me that cardiovascular disease and these are related heart is really is a challenge for our country, and all of us should come together to come out, give solutions to this particular problem, because premature mortality in terms of years of life lost because of the cardiac disease in India, it has increased tremendously in the last decade by I understand about 60% increase in the loss of life. This is really very uh, saddening, and I think the government should take and all the stakeholders work towards coming out solution for this problem. I'm sure our learned speaker, Dr. Chinmay Patra, who has years of experience on zebrafish developmental biology and organ regeneration, will give a good overview of this particular topic. And I believe the topic is very relevant for the PhD student, particularly, and other research scholars and scientists and clinicians who are participating in this webinar. I am told NUCSER has recently initiated two projects on familial uh, dilated cardiomyopathy and familial hypertension in collaboration with our medical school, particularly from the Department of Cardiology and General Medicine, where they are focusing on understanding of the genomic profile of the patients to identify the causative genes and functional characterization of the genes to the zebrafish models. Again, I should congratulate Anirban for this very proactive, wonderful areas he has selected for research. On behalf of the Nitte University, I extend very warm welcome to the guest speaker, Dr. Chinmay, and also Arati and Bharati, who are participating in this webinar. I congratulate NUCSER and the Consortium for organizing this webinar. This for a particular theme and particular dedicated cause. I wish you a great success for this webinar, and I thank the organizers for giving me this opportunity. Wish you all the best. May God bless you.
Thank you very much, sir, for your encouraging words. It's always a pleasure to have you with all the activities that we have, and we hope to have your continued support in the years to come. Thank you very much for your time, for your presence. Now, I would like to request Dr. Chinmay to start the proceedings. You can share your slides, Chinmay. While you do so, yes, I will introduce you to the audience.
Yes,
So, Dr. Chinmay is one of the few distinguished and accomplished scientists in the field of developmental biology in the country. Dr. Chinmay is a graduate from Jadavpur University in Kolkata. He is actually a graduate in B Pharma, followed by his MTech from IIT Kharagpur, his PhD from a prestigious institute in the world, Max Planck Institute for Heart and Lung Research in Germany, and then again 3 years of postdoc in the same institution, followed by his appointment as a scientist at Agarkar Research Institution from 2014. Chinmay has a lot of achievements to his credit. One of the few prestigious ones is his selection as a Wellcome Trust DBT India Alliance Intermediate Fellow from October 2019 to October 2024. He is also a recipient of the GSPSC Invitational Fellowship by Government of Japan for research in Japan from 2019 to 2022. He is also selected as the head of the Max Planck partner groups in India and awarded Early Research Career Award by DST in 2016 and Dad Fellowship for his MTech study in Dresden, Germany. And he is first rank holder in GATE 2005 in pharmaceutical sciences. That I didn't know, Chinmay. Many congratulations on that.
Thank you.
It's a great achievement, yeah. And he has received several grants. I think the list is pretty big, but I can only give you an idea. He has received grants over 6.5 crores Indian rupees so far. And his publications are all in prestigious journals. To name a few, Science, PNS USA, Blood, Development in Neurobiology, Biology, Cardiovascular Research, and so on. It's an absolute honor for us, Chinmay, to have you today amongst us and to listen to you. Thank you so much for your acceptance of our invitation, and we look forward to an exciting 40-45 minutes from your side. Over to you, Chinmay.
Thank you. So, good evening, and good morning, and also good afternoon. And first of all, I would like to thank Dr. Chakravarti and Dr. Professor Bandari for inviting me to give this talk and for such a wonderful introduction. So for today's talk, definitely I talk about heart regeneration and how we can use Zebrafish to understand that how we can promote heart regeneration in mammalian system. But before going to do that, I would like to spend a couple of slides to speak about that what exactly we are doing and why it is necessary in today's research. So whenever we talk about any, this basic research, and we always we think about it, the medicine or how the discovery of the medicine and all these things. But if you look at this before going to that at the medicine level, we should think about the pathological conditions. So it can be very simple, like hyperacidity, or maybe the superficial injury, or maybe internal injury like intervertebral disc degeneration. Of course, myocardial infarction is more traumatic, and the blood pressure, fever, and all these things. So whatever the disease conditions happen, what pathological conditions, that basically we start to think that what to do. We'll go to doctor, get medicine in the many different formulas, and it can be injectables or capsules, tablets, or in other forms. But we get certain molecules which help us, to get out from the pathological situation. So for every conditions, basically the drug or medicine we get, this having the small molecules which interact with its target. It can be receptors or other protein molecules or something else, but it must target something, or that basically the cause of these pathological conditions. So for any kind of treatment or discovery of the medicines, we must need to know that how these pathological conditions appear. So like for the hyperacidity, we need to know that how its stomach get more acid. In case of the degeneration, that how this degeneration happening and what are the processes involved in it. For this also the heart myocardial infarction, that how coronary blockage happened and why heart cannot regenerate. So we can think about it to improve it. So if we know the answer that how it is happening, so we can discover a medicine to stop that problem, to processes involved in pathological situation. And to understand this, scientists are using animal models that to answer all these questions. And when you think about the animal models, you always think about the mammalian systems like mice, rabbits, rats, primates, guinea pigs, and so on. But if you look at that, the history that we have more than 2,500 years back, we have started to use small animals to understand many things like in the sponge and also other small organisms. So most like the fruit fly having a big role that okay, we understand so many things using this small, the fly. Basically, in today's drug discovery, it has a major role. If you look at all these, the zebrafish is the youngest animal model organism we are using. For the last 3 decades, we are heavily using zebrafish as a model organism, and although it has been started around 50 years back. So if you look at the zebrafish habitats, they are from basically from our country, from India, Pakistan, Bangladesh, Nepal, and Bhutan. However, because of the pet shop, they are moving around the globe more than a few decades. And even in India and also abroad, you can find different colorful zebrafish. However, in the lab, we mostly use these gray stripes fish for the research purposes. So if you think about the research and development, and that is basically always you think and just for the benefit of the mankind, and relatively simpler, the system we use the cell culture because we can in vitro we can visualize each and every cells how they are behaving over the time. We can then move to the next step that is the in vitro tissue engineering or the organoids culture. So each having the cells and also extracellular matrix, and then we move to the in vivo system like the mice, rats, and the primates, and at last human. So it's a long path. It's like a path through the hill area. You can see the long distance, but nowadays one can make a tunnel and you can make shorter form. So I do believe that zebrafish having the this role that which can make this long distance research maybe make it a little bit shorter. And I can tell you that why I am telling like that. So zebrafish having the in vitro fertilization, so that means we can basically see that from the single cell stage to a complete organism development in vitro. And what I am showing here that if you I kept here that one male and female together, and you can see that small eggs are basically dropping out. So basically here the female is throwing the eggs and male is throwing the sperm, and within a fraction of seconds get fertilized and dropping it, and you can take it on the petri plant and grow in an incubator. And you can see what I am showing here, this single cell stage embryo, this one cell, and within 30 hours you can see that the developed embryo. They're very transparent. So here I'm showing for the example, this all the green cells you are seeing, these are basically blood vessels in zebrafish embryos. And of course that high fecundity, so we can get more than hundreds of eggs from one female every week. So that's why. So this seems this is a in vivo system, the animal model. However, we can observe at the cellular and the subcellular level in a living organism without killing the animal. So that's why I say that it has an advantage of the in vitro system and also is in vivo system. So we can use it for both the purposes. And nowadays we can also make the transgenic reporter lines and the knockout phase to study the function of specific gene using CRISPR-Cas9 or 10 technology. As I mentioned, they're pretty transparent. For example, since our lab works on heart, So I must speak about also art here. So this is the 2 days old zebrafish, and you can see here this black spot is one eye, and red it is quite it's showing the pericardial cavity. So here you can see the pericardial heart reside inside. So here I am showing that 3D projection of zebrafish heart from a transgenic reporter line where this all the green cells are basically cardiomyocytes, the only contractile cells. So there we have two chamber heart. You can see one ventricle, one atrium here. And if you look inside under this confocal microscope from a double transgenic reporter line, you can see two layer of cells at 2 days old stage of the fish. So outer green cells are the cardiomyocytes and inner one is the endocardium or endothelial cells. So it's the in-between ventricle atrium, one can see the atrioventricular channel. However, it does not have at this stage any, the valve leaflet. But if you look after 8 days, after 10 days of post-fertilization, you can nicely see this valve leaflet, two leaflets you can see over here. And also you do see that projections that which protrude inside the ventricle. So this is called cardiac turbidity. That is very important for the sense where the blood comes from the atrium to ventricle. It basically push the trabeculae and it gives the sense and to function for the cardiac and ventricular contraction. So also you can look under the microscope here and seeing the videos and the bright field image. You can see here nicely that how the atrium is receiving the blood and the ventricle is just throwing the blood through the outflow tract, or the because it is the blood is going to the gills forming region and throughout the blood. So we can observe the cardiac functions under this normal bright field microscope. Also we can look at the subcellular level. So you can see here from the light slit microscopy that is from the double transgenic reporter line, the red cells are showing the cardiomyocyte nuclei, and all the white is showing the filamentous actin. So one can see that the contraction and relaxation each and every cardiomyocyte, that how much extension is happening for a single cardiomyocyte in heart. So now it's all about that. Okay, this the beauty of the transparency of this zebrafish, that we can really look at the cellular and subcellular level during the development of the organ function. So the next question is that, okay, how much similarity between exists, because whatever we are trying to understand that from that with whatever the understanding will come from this zebrafish as research, that we can go back to the human and whether this knowledge can be useful for the pathological or physiological situations in human. So if you look at the genes, that around 70% genes are similar. So zebrafish at least have one ortholog of 70% of human genes, and also zebrafish having almost each organ except lung, what we do have. So that the development of this each and every organ and the role of these 12,700 or around 13,000 genes in organ development, and we can really study in zebrafish. If you look at the regeneration, zebrafish can regenerate almost each organ, and although we have the regenerative capacity for our skin or the liver, but most of the organs we have very limited regenerative capacity, where in contrast zebrafish having very tremendous regenerative capacity. So again, we can use zebrafish as a model organism to look at that natural regenerative processes, and also we can look at the function of these common genes, which is also in the human and the zebrafish, the function of these common genes in organ or tissue regeneration. So, if of course you should know that, okay, that's a little bit about the history of zebrafish research. So, zebrafish research has been started by Judge Rodriguez. Actually, he started around 1965 at University of Oregon, and he worked for around 15 and 20 years on zebrafish over there, and unfortunately, he lost him in 1984. However, in that in University of Oregon, his colleagues, they continued to work on zebrafish, and still some of them working at University of Oregon. But the next breakthrough happened in 1996, basically. So Christian Wilson followed from Max Planck Institute to Tübingen, Germany, along with 3 other research labs in USA. they conducted a DNA mutagenesis screening to identify the functions of very, a number of genes, and found a couple of hundreds of genes having the similar functions between the human and zebrafish. And in 1996, they have published as many papers from this work. And that was the breakthrough. And after that, you can see the look at the PubMed is every year is increasing exponentially. That the number of publications. And in 2022, more than 500 laboratories in around 35 countries working on jellyfish to understand many things like development, regeneration, toxicological analysis, and many. So I would say in every field now we are using zebrafish as a model organism, and in India more than 50 labs working on using zebrafish as a model. So now we'll come to the heart regeneration. So why it is important to understand the heart regeneration and why we need to talk about the heart regeneration. And I'd say that why I am fascinated, although I worked during my PhD and postdoc on heart development or cardiovascular development, but after studied lab in India, I thought to work on the heart regeneration, and this is really fascinating for me, and I do think that's very important. So why it is so important? So if you look at the heart, it's the people used to believe that it terminally differentiated organs means if you take the injury in the heart, it can't regenerate. But as many reports in recent years, in the last maybe 10 years, it suggests that it's not completely terminally differentiated organ, but it has very limited regenerative capacity. So over the year, our heart can regenerate. In mammalian heart, we can regenerate a bit, but the capacity is very limited. So what happens if any myocardial infarction happens? That means all of us would know that if the myocardial infarction happens because of the lack of blood supply, because heart muscle is very, it's always busy to working. It's like every minute it's pumping like around 70 to 100 times. It's contracting and relaxing, so it needs a lot of nutrients and the oxygens to function it. So like the any coronary blockage, what we are seeing in the green cells, these are basically coronary vessels inside the heart. I'm showing here the zebrafish heart. So if any blockage happen in this vessel, whatever the area is getting oxygenated blood and nutrients filled blood, it will not get the blood, and that's why basically that tissue will die, and that's lead to the cyanosis. And as soon as that the area don't get the blood, it's basically this cardiomyocyte, they do die. As I told that we have very limited regenerative capacity, so we can't really, our heart can't replace this dead cardiomyocyte with newly formed cardiomyocytes, but because due to protective mechanism, that dead cells get replaced with the scar tissue. So you can see the bruise is the plus scar tissue, and the big red is basically healthy muscles. So in one myocardial infarction, so one heart attack basically lead to kill around 25 to 30% of the cardiomyocytes. So that is 25% of or 25% of the cardiomyocytes replaced with only the scar or fibrotic scar. So heart functions gets reduced quite efficient, around like 25% reduction of the heart functions happen. So that's why we always tell after the third attack, people cannot survive because every attack kills around 25% of cells. So that means 70% cells to die after the third attack. So as I mentioned, that since we do have a very limited regenerative capacity, so now we need to understand that from that how we can improve this regenerative capacity. So that's why we need an animal model organism that can regenerate
Hello, Chinmay. I think there's a connectivity issue at his end.
like coordinates.
Chinmay, you there?
I can hear you.
Yeah. There was a bit of stoppage in between.
Now it's okay?
Yeah. Now we can see.
Okay, fine. Please let me know whenever some problems I can.
Yeah, sure.
So, yeah. So, you know, I can start now.
Go ahead, Chinmay. Go
Okay.
ahead.
So yeah, so that here what I'm saying that injured Zebrafish herd. So first you can see the footprint deposition. This is the big thing, and after that you can see this formation of the collagen spur. Collagen spur you can see here, similar to like the mammalian system, but it can completely replace the scar tissue with the newly formed cardiac muscles. So that again, the cardiac physiology is pretty conserved. At adult Zebrafish heart also beats around hundreds beats per minute. So it's quite similar. I should mention here, if you look at the rodents like rats and mice, their heart beats around 500 to 600 times per minute. So if you look at the ECG, it's quite different. This rodent cardiac physiology is quite different than the mammals. However, the Zebrafish ECG, if you look and compare with the human, it's quite similar. And of course that 70% genetic similarity, again I'm telling, so we can really try to use this model system, try to understand that how they can regenerate and the function of these common genes in heart regeneration. So our labs is interested mostly this extracellular matrix molecules. So the molecules that's basically synthesized in the cell and secreted outside the cells. So earlier, actually people believe that historically the matrix tissue or extracellular matrix basically inert scaffold. They are only the supporting materials, but the recent year studies support that extracellular matrix basically is a hub of many important molecules which help in the regeneration and also tissue homeostasis. So today's work I'm going to talk about this. Last year we published in development, and this work most majorly done by Devanjan, who is now doing his PhD in Goethe University in Frankfurt. So what he has done, he basically hypothesized that secreted molecules or extracellular matrix molecules, they are induced means shows the increased expression in injured or regenerating Zebrafish heart are associated with the heart regeneration. So that means they have positive role in heart regeneration as Zebrafish heart can regenerate. So he has basically isolated, this RNA from the healthy heart and also the injured heart, 4 days injured heart, and identified the couple of molecules which are induced. Among them, CTGFA and CTGFP, those molecules he found that pretty interesting to look that okay, why they are going upregulated over here. So if you look at the CTGF, that CTGF in human only one gene. However, in Zebrafish there are two paralogs, CTGFA and B. And if you look at the structures, the structure is quite similar. The amino acid sequence is quite similar with human. Here it is HS CTGF means human CTGF, and you can see here means Zebrafish CTGF and Zebrafish CTGFB both are quite similar at the amino acid level and also the domain structure wise. So that is around 38 kilodalton protein, and it has 4 cysteine domains. So since they are quite similar at the amino acid and the domain structure, so we can it's most likely that Zebrafish CTGF having the similar role like the human or mammalian CTGF. So first what he has done, he looked for the expression level. So when you want to know the function of gene, we need to know where does it express. So first we check the CTGF A and B because Zebrafish is having the two paralogs. So what we observe that CTGF A expression is much higher. You can say more than 10-fold higher than the CTGF. So it's thought that okay, that means CTGF has the most prominent function in the heart regeneration. And when we perform this RNA expression, so what we have shown here, the injured heart sac cells, and CTGFA is the RNA, whatever the blue dots you are seeing that are CTGFA RNA, and MF20 is the cardiomyocyte. So you can see the healthy region is the red, and in the bottom one you can see the uninjured region is the red here. This is the cardiomyocyte, live cardiomyocyte. But in the injured tissue, you can see the bottom one, where you do see the mostly that RNA expression, the blue indicating the CTGFA expression. If you look at the bottom one, that CTGFB expression is very few, relatively less. So that CTGA expression is induced in the injured tissue. Now we wanted to know that, okay, which cell types basically express. So what he has done here, generated a reporter line, transgenic line, where EGFP expression was under the control of the CTGFA promoter. So in this transgenic reporter line, he has again made the injury in the heart at 26. So what we can see here, this image is showing the bottom one is the injured part and green indicating the CTGFA expression. So you can see the injured tissue region, that the green cells, but uninjured region is on, there is mostly this green, CTGF expressing cells are missing. The next panel here, this red cells, basically these are the endocardial cells or inner lining of the heart. So that is visible throughout the injured and uninjured tissue. Or you can see that all these green expressing or CTJP expressing cells in the injured tissue, they overlap with these endocardial red cells. So that means CTJPA basically expressed in the injured endocardial cells. Bottom panel here we are showing that this tropomyocytes is a cardiomyocytes. So you can see the healthy region again, the cardiomyocyte, where we do not see the CTJP expression. However, on the bottom, the injured tissue, where all the cardiomyocytes are dead, there we do see the this expression of the CTGFA. So what we found altogether, that CTGFA expression is basically induced and it expressed in the injured in the endocardial cells of the injured heart tissue. So then we look at the CTGF protein localization. So we have done the immunostaining. So here what I am showing is injured, the heart sections through the injured ventricle. On the bottom, it's showing the injured area, left side, and you can see that all the red cells, the cardiomyocytes. So again, when the zoomed image, you can see here CTGF expression in the injured or localization of the CTGF protein in the injured tissue mostly, and also adjacent tissue. However, we do not see too much expression in the uninjured tissue. So it supports that CTGF is probably having the role in the regeneration. So understand what he has looked at. That he has looked at the CTGFA mutant because we're going to know the loss of function of this gene. So if you remove the genes, what happened with the heart regeneration? So he used this mutant element, CTGFA mutant, the mutation actually at the VW domain. These animals has been generated with the help of tannin technology. And after the when we have done the we took basically wild type and the mutant animal, and for our study we used the cryoinjury model. So we open up the chest, and here image is showing this. You can see the heart, and we make the injury with a cryo probe. So what what it has done? He used the cryo probe. It's nothing. Here is showing like the cartoon is showing that it's basically copper wire. And you need to just put it to the liquid nitrogen, that make it super cold, and then you open up the chest, and that ventricle will pop up and they touch with this super cold probe. So basically it makes the cryo bar. So I'm here showing that the Ganesh has done this one heart injury model. So I can show you that how we do this cardiac injury. We need very simple. So he put this adult Zebrafish into the Triton solution to anesthetize it, and these are the simple instrument you need. One forceps, three forceps you need the scissors, and this is the cryo probe. It's simply that copper wire we put into the liquid nitrogen. Now the sleeping fish you put on sponge having a little bit of water, so it should get a little bit of oxygen for the survival. So now he is removing the scale from here. And then you can see this, he will just opening up the chest region. You can see now that the ventricle is, you can see the beating cardiac ventricle. Then now we'll touch this super cool cryo probe on tip of the ventricle. So it will be a couple of seconds touched with that, and then it's simply released. So you can now just simply put this together and put into this cleaned water. So is within a minute it will start to swim, and basically he will, you can see, started to move. So yeah. So then after we keep this animal in the system, and then after 60 days we keep them for 60 days and look for whether regeneration happens. So what he has done, he has used both the elements. Here you can see the white type element that make the sections through the cardiac ventricle and stained with the phalloidin, stained all the cells, whether it is cardiomyocytes or non-myocytes, and MF20 will stain only the cardiomyocytes. So the yellow area, this is basically the injured area. So you can see this injured area, cardiomyocytes, they are already filled, the injured area, but you can see that they are not as good as the, sarcomeric structure as like the healthy area. So here orange color is showing the healthy area of the heart of this wild type animal, but you can see that is filled with the cardiomyocyte, the injured tissue. So that means regeneration happens. However, if you look at the mutant, this injury area, you can see this no cardiomyocyte over there. Very few cells, this covering the outer layer of the heart to protect it, and it's only, these cells are present there without any cardiomyocytes. However, in the healthy area, you can see similar to like white type, you can nicely striated cardiomyocytes. So next we ask that whether, is there the scar tissue. So what he has looked at again in the wild type, heterozygous animals, and also in the mutant. So what he observed that in this, this injured tissue of the wild type animal, you can see this very limited, little bit of the scar or collagenous scar. However, in the head is more, and also the mutant is more. And even after this is after 2 months, even if you look after 5 months, and also you do see the scar tissue, and this is the cortex. So that means that in the absence of the CCNPA, that Zebrafish are they fail to regenerate, and they basically end up with the long-lasting fibrotic scar. If you think about any kind of injury, the first process is the inflammation. So we do see the inflammation, and after that, basically this open tissue has to be closed, so that is the scarring, and the scar tissue has to be replaced with the functional tissue for the functional regeneration. And that for the scarring to this, regeneration, it's many things needed. One is the angiogenesis, of course, we need the blood vessels will enter there, and the functional cells, like you need these skin cells for that you need the cell proliferation, and of course those cells will proliferate in the healthy region, and they have to migrate into the scar tissue, and that's the way it is basically replace this scar tissue with the functional tissue. So that's why you look first at what happened with the coronary vessels formation or blood vessels formation in the heart and the cardiomyocyte movement from the healthy region to the injured tissue. So interestingly, what you observe basically, we used here double transgenic line with that the TS set is the whole red nucleus, there is the cardiomyocytes, and you can see the blood vessels are in the green. So you can see this healthy region full of cardiomyocytes, and in this injured tissue area, you can see the few cells already entered in around after 4 days of injury, and all these cells basically they are attached with the coronary vessels or blood vessels. These blood vessels also entered into the injured tissue. So that means that coronary vessels basically guide the cardiomyocytes to enter inside. Surprisingly, what you observed here, interestingly, that in the cardiomyocyte CTGF mutant, although they have plenty of coronary vessels in the injured tissue, but they do not have any cardiomyocytes. So that means that cardiomyocytes are driven by the coronary vessels, but it required the CTGFA. Without CTGFA, even coronary vessels cannot drag them into the injured tissue to replace the dead tissue with the functional cardiomyocytes. Dramatic effect you can also see after 12 days of injury. You can see in the wild type scenario that massive number of these cardiomyocytes they migrate into the injured tissue, which in case of the heterozygous and also the mutant, it's basically pretty less. So we found that CCNPA is very important for the cardiomyocyte migration. Next, we look for the cell proliferation because the cardiomyocytes need to proliferate to fill these gaps, not only the migration. So we check whether in the CCTGFA mutant they have enough number of proliferating cardiomyocytes. So what we have done, we basically pick the current injury, and then injected EDU. So EDU incorporation will tell the proliferating cells or the cells which going through the DNA synthesis, and then after 24 hours he has isolated the heart and performed the staining. So here again, we have used this transgenic reporter line where the red cells are basically nucleus of the cardiomyocyte. So you can nicely see here the healthy area and also the injured tissue, and every green dot showing the proliferating cells. So the red cells, if they are green, so that means proliferating cardiomyocytes, and blue they are showing basically the nucleus of all cells. So if you see the wild type, here you can see the many cells are yellowish, so that means this is the red nucleus, they are also positive for green, with the proliferating cells, but in case of heterozygous animal and also mutant, we do see less number of the cardiomyocytes. So it is telling that CTGFA is required also for the cardiomyocyte proliferation. So what we have shown so far, that connective tissue growth factor A is important for the cardiomyocyte migration and as well as proliferation, and most likely it's help in the heart rhythms. But how it is happening? So to understand the process, that how this TGFA is regulating this heart regeneration, what Devanjan has done, he has performed the transcriptomic analysis. So he has isolated the RNA from wild type injured heart after 4 days of injury, and also CTGFA mutant heart after again 4 days of injury, and then performed RNA sequencing and identified differential expression. Surprisingly, what we found that only 7 genes are differentially expressed, and where mostly that fibronectin and collagens are downregulated, and you will see that one CXCR3.1, this is a typical couple receptor, chemokine receptor, that is basically upregulated. So since so we have looked at this transcriptome level. So transcript level, so we check that whether what happened in the protein level. So we check 2 molecules, one is the fibronectin and another is the collagen PA1. So in the fibronectin, what we did after 7 days of injury, what he has done that he made the sections through the cardiac ventricle and the outflow tract, and what he observed that fibronectin is nicely localized in the outflow tract. You can see the double star, and also throughout the ventricle and also in the injury site. So you can see the fibronectin expression over here. However, when you look at the mutant, fibronectin expression in the outflow tract remain unchanged. So you can see this comparable to the wild type heart. However, when you look at the ventricle of the mutant injured heart, the expression is pretty less. And again, the similarity. When you look at the collagen PA1A, it's nicely localized in the injured tissue, which is missing in case of the CTGFA mutant, and this is the quantification. So this image is again telling that CTGFA mutant, they basically fail to they express this collagens, fibronectin, and that's CXCR3.1 molecules. So we know that is TGF beta pathway involved in this extracellular matrix molecule T2. So we checked what happened with that TGF beta SMAT3 pathways. So the readout will be that the phospho-SMAT3 localization in the nucleus. So what he has done, he has again the make the injury, and after 7 days, we looked for the SMAT3 localization. So here the phalloidin staining, which will demarcate the uninjured tissue and also the injured tissue. So this in the injured tissue, a massive number of cells which have been phospho-SMAT3 positive. However, in case of the mutant, is dramatic, decreased expression of this phospho-SMAT3. So this is the quantities. So what we have seen that in the CTGFA, basically somehow regulating that TGF beta, signaling, and we don't know how. And this signaling regulates the same molecules involved in the heart rhythms. So to understand that, how it is happening, so we checked first the transcript level, whether the TGF beta, but we do not see the transcript level change of any TGF beta. However, when we look at the TGF beta 1 protein, we do see that expression of TGF beta 1 in the injured tissue, injured heart. You can see here the healthy region and the injured tissue, but which is decreased in case of the CTGFA mutant, and this is the continuous. So what we have identified that CTGFA somehow regulating TGF beta viability, but we still don't know that how this is happening, and which leads to basically TGF beta phospho-SMAT3 signaling and ECM molecule expression, and which led to cardiomyocyte proliferation and cell lines. So now how it is happening with it too. So it's two things we have shown, that one is cardiomyocyte migration and the cardiomyocyte proliferation. So first, that in the earlier study has shown that TGF beta is important for the cardiomyocyte proliferation, but we did not know that about the role of TGF beta signaling in cardiomyocyte migration. So what Devanjan has done, he made the cryoinjury and they treated this animal with an inhibitor which inhibit this TGF beta SMAD3 signaling until from day 1 to day 6, and then look for the cardiomyocyte migration. So what is this again, this transgenic animal? You can see the healthy region having the cardiomyocytes and the injured tissue. After 7 days, you can see the plenty of cardiomyocyte already appeared there. But in case of the CSP treated animal, we do not see that cardiomyocyte in this region. So what we observe that CTJ basically regulate TGF beta biology and which lead to the CCA molecule expression, and somehow this is regulating the cardiomyocyte proliferation migration. So since we know that CCA2 is required for this cardiomyocyte migration, proliferation, and also heart regeneration, so there was an ask that whether if we give extra amount of CTGFA, whether we can augment the regenerative process in Zebrafish heart. So used transgenic lines where basically CTGF is expressed under the control of Hitchcock protein 70 promoter. So HSP70 promoter actually remain inactive in Zebrafish because we keep Zebrafish at 28 degrees centigrade water, and HSP70 promoter get activated at around 37 degrees centigrade. So in normal condition, it will not make this extra CTGF. So he made the injury, and either with this background, this transgenic, or without that. And this is the wild type scenario, and then this is the HSP70 CTJ. Both are wild type, but this animal having the HSP70 CTJ. And he gave it shot for 7 days, and have to observe that upon the extra amount of CTJ supply, you can see that increment of the cardiomyocyte proliferation. So cardiomyocyte proliferation, proliferation can be induced by giving extra CCA2A, and most importantly, when if the hit shock, this extra amount of CTGF supply for a month, what we can see here, you can see the wild type, the scar tissue is a lump of collagenous scar, but you can see in the that the animal which received the extra amount of CCA2A, that this cardiomyocyte basically entered inside the scar tissue, and basically it helps in the regenerative processes. So that means that the CTGFA is required for the heart regeneration, and it can also augment the regenerative processes. So what we have shown that connective tissue growth factor is basically expressed in the endocardial cells at the border zone of the injured tissue of Zebrafish heart. It somehow regulates TGF beta viability. We don't know how it regulates, but we have seen that less amount of the TGF beta 1 protein, and that's why basically less amount of this ACM genes expression, and which led to CM migration and proliferation, and the scar regression, and with the heart regeneration. So with all this, I would like to thank to all for listening me, and my collaborators, and the lab members, and all the funding agencies. I'll be happy to receive questions and answer.
Thank you very much, Dr. Chinmay, for your wonderful presentation and for taking us through this CTGF story and how it is contributing to the process of heart regeneration. We do have a few questions, but before I take those up, I would like to request Dr. Aarti or Dr. Bharti if you have any questions to ask Dr. Chinmay.
Sure. This is Aarti. Can you hear me?
Yes, please.
Excellent. Okay, so Dr. Chinmay, that was a beautiful talk. You really, gave all the controls and very convincing data that your model of regeneration is truly one to be watched and looked for in not just now but also in the future. So I have a very fundamental question for you, which is probably something that that you have considered in the past, and others have too. Why have humans lost the capacity to regenerate their hearts? What is it from an evolutionary point of view that makes the human heart different from the lower animals like geckos and others that have the capacity to regenerate organs? Our livers, for example, can regenerate themselves, but why has the heart lost this capacity? That is question number one. And question number two that I have for you is that do humans have the equivalent of a CTGFA gene that has either lost its capacity to function or altered its capacity, or has other isoforms that are somehow regulating it in a way that is different from zebrafish? So please.
Yes. Yeah, very good. Yeah. So my personal view is like that. So yeah, we do have we've not completely lost the regenerative capacity. Even now, recent study we can see that even neonatal rats and mice immediately after birth they can efficiently regenerate their heart. So we do have to that we have the regenerative capacity, but over the time, like basically it's get reduced and will come is come to the bottom level. That when we have the massive regional injury, we can't do this. It could be that two reasons. That one is that since it's a very hardworking organs, so immediately after injury is basically it start to repair it, because if this regeneration process take a long time, so it's repair. But during this repair mechanism, probably the scarring, whatever happened, this may go beyond that what exactly is required, or which basically maybe to some extent inhibit the regenerative process. One thing. Second is that for every regenerative process, we have to induce the tissue to de-differentiate. So if the cardiomyocytes remain it's in the normal stage, at the functional stage, if it does not de-differentiate, it cannot regenerate, or it cannot make the new cells and go to the injured tissue. So probably that is another thing, that okay, somehow our cells also don't go to that extent, that de-differentiate and to make the daughter cells which take care for the regenerative processes. So this is my understanding, and that's why we don't have efficient regenerative capacity. So we need to play in that area. And then that CTGFA. So now it's called CCN2 gene. We do have also CCN2 or CTGF. Zebrafish having two, CTGFA and B, and human having CCN2. And indeed, in after myocardial infarction, CCN2 goes up, and that's why people believing that CCN2 is basically inhibit the regenerative processes, because it goes off and scarring and everything happening. But in fact, it is not like that. So it could be that I do believe that it's a regenerative response, but due to some reason, heart failed to carry it. So that CCN2 goes up and try it, but it fails. So that happens in the heart. And we are working on another organs, and where basically we do see the similar the tendency that if you give the CCN2, we can also promote even aged zebrafish. We can promote the regeneration without making any external injury. So if you have aged the organ and having the less regenerative capacity, and if we give extra amount of CCN to it, you can even induce over there. So yeah, this is actually suggesting that it is having the role, but somehow failed in human.
Thank you. That was very enlightening. But I'll allow I won't take up more of your time. Let others ask you questions too. Thanks a lot.
Thanks, Swati. Dr. Chinmay, I have a few questions for you. One question
please.
from the participant is it reliable to use zebrafish to try new human drugs?
Yes, actually, there's a lot of labs are now doing this, this small molecule screening using Zebrafish. So even directly go through any other animal models, we can our discovery from the Zebrafish embryo and the adult, we can take it to the bedside because there are lacks of FDA approved drugs. Uh, you can basically screen on zebrafish for any kind of disease, and if we get the fruitful result, one can directly take it to human without going through even all these channels if they are FDA approved molecules. And that's why in the personalized medicine, uh, actually this kind of screening helped a lot.
Okay. There's one more question. I think I could not understand the question clearly, but I think the participant is asking: is there any specific stain that can selectively, stain the heart in fish?
Yeah, this is actually report line, so we can use any cardiomyocyte specific or actually tissue specific promoter, and in front one can give the green fluorescence protein or red fluorescence protein, and that tissue basically show the green fluorescence expressions or red when you put it in under the UV. So this is it's mostly I use the transgenics whatever I've shown, and
So there are a few more questions. I mean, a couple of them are from my side. But so Chinmay, the mutant that you developed, did you develop on a transgenic background, or you crossed with a transgenic line to get a dual, situation?
We have generated it just like normal, white tape, without any transgenic background, and we just cross it with the different transgenic as per our requirement.
Okay. And then how did you identify the homozygous based on
So we use
genotype?
by genotyping.
Genotype. So this CTGF null mutant that you have developed, does it have any other phenotype, like say elevated lipid profile or something?
It has phenotype, and we are going to communicate.
Okay.
So maybe next time we'll talk about it. So yeah, that's very interesting, phenotype, and yeah, I'll talk about maybe in 3 weeks.
And these are viable as
They are
mutant?
viable. They are viable mutant.
Are they sexually active in the sense? Are they?
Yes. They are fertile.
Okay.
They are completely fine. Means they are fertile. They can swim. Everything. Okay. And
Also, one quick question: are these are the knockout mice? Are the knockout zebrafish of CTGF? Are they CTGFA? Are they shorter lived? Do they live shorter life?
No.
Oh.
They do live normal. Since zebrafish having two, CTGF A and B. So mouse mutant, global mutant, they do die. CCN2 or CTGF mutant, they do die, but zebrafish not. So it's most likely that the CTJB actually what we observe that it goes up around fourfold up. So maybe for the development that CTJB take the role in the absence of CTJB, and that helps it because yeah. So that is the things we don't have any proof for that, but we do know that their CTJB goes up.
So you're saying some sort of compensation happening.
Compensation happening. Yeah.
And another, it's a very general query. So you have done a cryo injury to your model, isn't it? But the kind of injury that we see in a myocardial infarction is not a cryo injury in the sense it is based on, shortage of oxygen supply to the heart muscle. So how do you find the similarity in that?
Yeah. So, if you up to the injury, we don't have similarity. We are making the cryo burn, but post injury, if you know that after the myocardial infarction, basically that tissue, the whole bunch of the tissue that die. Same thing, the cryo injury basically kill and the whole bunch of tissue die. Their dead, every kind of dead cells are there, as it also in case of after post myocardial infarction. So yeah, that I say that is closer, but yeah, it's not exactly same. Of course not, because we make the cryo burn. It is not death due to the lack of oxygen and death due to the
It's a physical injury that
physical injury. Yes.
Right. Okay. I think there are a few more, but I think we are almost 10 minutes beyond our scheduled time. So I think we'll stop at this, and we hope to have Dr. Chinmay sometimes come over personally to Mangalore and maybe talk to our students and perhaps teach us a few things. I do know that one of my students went to his lab to learn a little bit about CRISPR technique, and she has been successful in developing a mutant. We are in the process of analyzing it. As you said, we also found something interesting in this mutant. So we look forward to your visit personally, and it is always better to have in-person interactions than over the internet. But anyway, thanks for your time, Dr. Chinmay. I really appreciate your kind acceptance of our invitation and for giving us such a wonderful insight into your research. And I'm sure those who have attended the session, they are immensely benefited from your talk. I also thank my university for giving us this platform to organize such webinars, for giving us this purpose of getting into basic research. And NUCSER, as our Honorable Vice Chancellor mentioned, it is an institution that was started with an intention of, promoting basic research in a medical university. So I think we are in the right direction, and with support from accomplished researchers like you all, I think we have a long way to go, and I'm sure we will not make the university feel disappointed. On that note, I think I thank my Vice Chancellor for his time, for his participation. I also thank Dr. Aarti and Dr. Bharti for being here with us. And I also thank Girish, our IT assistant, for helping us with the live streaming on YouTube. And finally, I thank all the participants who have registered for this webinar and who have joined on YouTube. The seminar will be available on our channel, Nitte channel. One can look into that at any number of time to know more about it. And finally, Dr. Chinmay, thank you once again. We will look forward to your continued support as a researcher, as a good friend, and as a Zebrafish, community in India. On that note, let me conclude this webinar. Once again, thanks to everyone for being here with us. Thank you very much.
Thanks, everyone. Thanks, everybody, for listening.
Thank you.
Thanks a lot. Yeah, it was excellent.
Thank you.